Yeah, thank you for the introduction. I will try and get through all this. If we start to hit the time and people start leaving, I'll cut a little bit out at the end. I'm mostly going to talk about my PhD research and try and give some pretty general background into why this topic might be interesting to people and why it's been interesting for hundreds of years. And then I'll give you my contribution to it, and at the end I will tell you a little bit about what directions I plan to go with this. So we're talking about statistical model choice in historical biogeography. And first, we should talk about what historical biogeography is. And you could imagine various definitions, but this is a pretty simple one, like how things got to be where they are. It's basically the question you want to answer in historical biogeography. We know things are living in various places on the planet, and it would be interesting to know why they have the distributions they do. So can anyone guess what this is a history of? People who are in the field will sort of like, oh. Sorry? Yeah, it's human. It's the sort of history of human migration. So we started off in Africa here, and that's the ancestral homeland for the modern human species, and spread out through Europe and Asia, and then into Oceania and into South America. This sort of picture there's a lot of different kinds of evidence for it, but one of the big ones was, here's the sort of key for it. This is just over the last few 100,000 years. This is based on mitochondrial DNA trees, but you can do a lot of different kinds of evidence for it. This is if you do allele frequencies and do sort of a distance measurement between populations, you get this sort of pattern. You get essentially what's interesting is that populations, if you force them to cluster, they'll cluster by space. Basically, human populations that are closer in space are more similar genetically. Everyone's very similar genetically, but there's a little bit of variance is explained by the geographic pattern. And what's interesting is if you look at the sort of genetic distances between populations and you rotate the map a little bit, you can basically get the geographic pattern in the map of genetics. So you get sort of America at one end and Africa at the other end and Oceania at the other end. And this is just evidence for the idea that people don't move everywhere every generation, right? They tend to reproduce and breed in a local region, and genes generally slowly spread around across the population. We go to the complete other side of biogeography, the other end. We've got sort of the phylogenies. In this case, there was a science paper that redid Wallace's biogeographic regions. Alfred Wallace developed the first big biogeographic classification of the planet. And these guys took phylogenies of thousands of mammals, thousands of birds, and thousands of amphibians, and then looked at how those clustered geographically in space. And they developed, they sort of recovered Wallace's basic regions with a few refinements. They sort of devised these major biogeographic provinces, which tend to be, essentially based by continent, with a few more intermediate regions. And they did some interesting clustering of distances between regions here. To sort of get a sense of, this is a non-metric multidimensional scaling plot. Basically, you take the matrix of all the distances in phylogenetic space between all these different regions, and you flatten it to 2 dimensions, and you get sort of a distribution of the relationships. This is a 2-dimensional representation of the distances between all these different regions in phylogenetics, in terms of phylogenetic similarity. And what's interesting with this is when you rotate that around, right, you can get something pretty similar to modern geography too. So instead of just being like current geography for modern humans over the last 100,000 years, we have, most of tetrapods across millions and millions of years, and you get a pretty similar overall geographic pattern in the sense that essentially phylogenetic relationship correlates. It's not perfect, but it correlates with geographic relationship. Things that are close together on the map, on average, are more closely related. So these are just some big patterns in biogeography, and this is the kind of thing, this sort of relationship between phylogeny and evolutionary history and geographic history is part of what has made biogeography interesting for a long time. And we can go right back to the origins of evolutionary biology, right? Before the origin of species, Alfred Russel Wallace, co-discoverer of natural selection, he wrote a paper in 1855 that suggested that every species has come into existence coincident both in space and time with a pre-existing closely allied species. And this was a puzzling thing if you thought special creation was true and organisms were just poofed into existence, right? Darwin took this up. He actually cited Wallace's paper in The Origin of Species, and Darwin summarized his biogeography analysis in a couple of chapters. He said, the more nearly any two forms are related in blood, the nearer they will generally stand to each other in time and space. So these are the big observations, and this was one of the big pieces of evidence for the theory of evolution in the first place, was that it looked like organismal similarity or phylogenetic relationship correlated with geography, and this was simply because things take a while to move around. Moving long distances is rare compared to moving short distances. So evolutionary theory in the beginning, one of the things it solved was the basic question of why things are distributed where they are. You know, basically they move around slowly and they tend to stay where they are. But this, when evolutionary theory was sort of erected, this created some new puzzles. And one of the big puzzles was often you do have fairly closely related groups. This is southern beaches from New Zealand and Australia and South America. They have these widely disjunct distributions. They're living, this is almost, you know, these South America and Australia are opposite sides of the Pacific Ocean. It's almost halfway around the planet, right, to get around from one to the other. And how do you explain a distribution like this? And the two big explanations are essentially vicariance and dispersal. And not everyone knows what vicariance is, so I feel like I should mention that. Vicariance is simply the idea that the ancestor of a couple of species had a widespread distribution. It lived in multiple places. And then some barrier came up and split the species into two, and eventually they developed into two independent species. So that's what we talk about when we talk about vicariance. Dispersal is simply the idea that the ancestor lived in one region and then jumped to a new region, or at least the range expanded, and then later broke up. And there's a couple of versions of dispersal. But vicariance and dispersal are two ways to explain distributions where things live in fairly different places. Having, you know, a beech tree which has a limited seed dispersal capability disperse across the Pacific Ocean, for example, seemed surprising and doubtful to a lot of biogeographers. So one of the ways that people attempted to explain this was by postulating geological land bridges. Back in the 1800s, people knew that the ocean had risen and fallen. You could see marine sediments on land. So it was at least plausible that maybe sometimes the ocean floor came up and there was dry land and organisms could disperse across it that way. This was all sort of before plate tectonics. And there was a lot of discussion. You know, people assumed the continents were fixed, but Darwin himself was actually skeptical of these land bridges because it became sort of a thing where anytime any biologist observed a closely related group, like a genus living in two different places, they would postulate a land bridge. And so you sort of had people just, you know, sticking land bridges everywhere throughout time. And you could see why that would be a problematic explanation. And Darwin actually, here he's actually ranting and raving to his friend Hooker. And he says, I must try and cease being rabid and try to feel humble and allow you to make continents as easily as a cook makes pancakes, right? So because Hooker was proposing land bridges, and Darwin's like, you know, at some point, basically this becomes an unparsimonious explanation if you're just going to sort of tweak geology endlessly to make it fit your distributions. So Darwin was kind of a dispersalist, and his friend Hooker was kind of a vicariance person. And this debate has continued since then. With plate tectonics, we had a potential solution to problems like this one, especially in the southern hemisphere, because the continents were linked up back in the past. And so if groups like southern beaches are old enough, they could be explained by just riding around on the continents. And this, you know, plate tectonics became dominant in the 1950s and 60s. And following that, vicariance biogeography really became the dominant paradigm in biogeography. However, in the last 15 years or so, vicariance biogeography has come under attack. And there's been a big push back towards the dispersalist side of the argument. Basically, a lot of these molecular dating studies have been done, and they've been increasing in rigor. And a lot of times when you do a molecular dating study and look at the divergence times of species within groups, their divergence times are much too young to be explained by plate tectonics. So that kind of, if you accept the dating, it forces you to think that dispersal happened for some of these widespread groups. And so here's a couple of papers that made this argument, resurrecting oceanic dispersal, long-distance dispersal, really rare events, which you wouldn't think, you know, a tree could get across the Pacific Ocean, but maybe once in a million years, a tree seed falls on an iceberg, and the iceberg, or a glacier, and the glacier goes into the ocean, and the ice wraps across the ocean, whatever. These things are incredibly unlikely, but maybe on the scale of millions of years they actually happen. And there's a lot of, Darwin and others have studied, there's a lot of ways to get things dispersing long distances. So dispersal's had a recovery, but what I found interesting when I was getting into this field was that this debate was unresolved. It's sort of an eternal debate. Here's the vicariance people have been pushing back, and they have some particularly colorful quotes. Here's a book from 2012 published by University of California Press, like across the street from my old building. This guy says, most modern biogeographers follow Meyer in accepting that allopatry can be found by vicariance or founder dispersal, but I'm only going to accept vicariance. He says, I'm just going to rule out founder event speciation. It's his method throughout his 500-page book analyzing the biogeography of hundreds of different groups. Another paper, or another quote from this guy, founder dispersal is controversial and may not exist. And the reason people don't like founder dispersal is it seems like it's kind of an ad hoc explanation. You can just postulate a rare event and get things distributed around the planet. And you could sort of explain anything that way, right? If you're willing to say, well, once in a bajillion years, you have some freak event, some storm, tornado picks up the frogs and moves them across the ocean, whatever it is, and all these things have been proposed. Maybe that's not scientific. Maybe the only thing that's scientific is using vicariance as your explanation. So that's the argument back and forth. And my summary of this is kind of like this. Once you're into the historical biogeography literature, you get tired of this debate, right? Because it always comes back to these issues, and you're just like, oh God, not this again. And so I resolved to swear off this debate. But then when I got around to finishing my PhD, it became apparent that it was incredibly relevant, and it's sort of the basis of why we are where we are in historical biogeography and what we need to do to improve things. So that's the historical background. The basic procedure that we use when we do analyses in historical biogeography is we start with a phylogeny, and you get that from DNA or from morphology or some combination. We get the geographic ranges of the different species in a tree, so we know where they live now. We assume that there's some process, vicariance, dispersal, that explains their distribution. And then we conduct inference, and we try and infer down the tree what the history is. Where did the ancestors live on that tree is basically what we're going for. And this can occur under different frameworks, parsimony, maximum likelihood, or Bayesian. And actually, a lot of people focus on these different statistical sorts of inference methods, but they're not actually the most important question. The most important question is really what model are you assuming? What processes are you going to allow? So I tend to work in the maximum likelihood and Bayesian framework, but a lot of what we're talking about could be done under parsimony as well. So I should review briefly the methods that are in use in the literature amongst historical biogeographers. There's kind of a couple of main ones, which I'll take you through briefly. Character mapping, which is just treating biogeography like any other character. One pretty famous method is DIVA. It's by Ronquist. It's dispersal vicariance analysis, so it tries to mix dispersal and vicariance. The most popular thing right now is probably Lagrange. It's a likelihood method that's sort of like DIVA. It allows different ways of moving species around the planet and uses a maximum likelihood method. There's a new method that's a paper that I was on that's published this year called Bay Area, which I'll talk you through in a little bit. And these methods, they have different sorts of statistical methods. They all use phylogenetic information. You can sort of see some of them are quite popular. They've been used hundreds and hundreds of times in the literature. And this is basically what exists. There's a few other new things that aren't very widely used yet. So we'll take you through these methods here. Method number one is treating biogeography as just a discrete character on a tree. So often, if you're interested in tracing the history of different ovule positions in flowers, right, you could put the characters at the tips of the tree and run some inference framework and get the ancestor states. This is a pretty standard procedure in phylogenetics. And you could do the same thing with biogeography. You just put the geography on there as a character, and you make an inference. The weird thing you're assuming when you do this is that you sort of assume that your biogeography changes states suddenly. If the ancestor lives in region A and you infer a switch to region B, you're sort of assuming that along a lineage you had a sudden switch from A to B. right? So the species picked up, left wherever it was, and jumped over to a new region, and left no trace of itself back in the old region. That's a kind of a peculiar model, right? So this is the difficulty people have with using just standard character mapping on biogeography. So the sudden conversion event. The nice thing about it is you could do a cost matrix for parsimony, or you could do a rate matrix and infer the rate at which these switches happen. But it's always bothered people that you're sort of assuming this instantaneous range switching thing. There's even more sophisticated Bayesian versions of this where you have perhaps different frequencies of occupancy of different islands, and you have different rates of basically dispersal between islands. But this is still all assuming that everything can only live on one island, and that when things switch, you go from A to B suddenly, and you don't leave any ancestors at A when you do that. So this is still a popular method, but it is kind of peculiar. Method number 2, it overcomes some of these difficulties. The DIVA method is very popular in biogeography because it doesn't make the assumption that everything has to live in only one area, and that ranges switch suddenly by kind of just ditching your ancestral range and jumping to a new place. Dispersal vicariance analysis, it allows species to live in single areas or in multiple areas. And the way it moves things around is with dispersal events, where here it means really a range expansion. So a species living in one area has a dispersal event and expands to live in 2 areas. Or you can have an extinction event and go the opposite direction. That looks like this. These are anagenetic events along branches, range expansion, range contraction. The other nice thing is it's not just a dispersal explanation. The DIVA program has a vicariance explanation, where if you have a widespread ancestor, it can break up and produce descendant species in subsets of the range. So, and DIVA assigns, it's a parsimony method, it assigns a cost of 1 to dispersal and extinction events, and a cost of 0 to vicariance events, so it really actually tends to prefer vicariance events. And there's a pattern in DIVA analyses where if you have a bunch of species living in individual regions, you will typically infer the ancestor is living in all the regions. And then just the ancestor breaks up and produces your distributions of your modern organisms. So these are the kinds of, you can see yes and no, the sorts of processes that they allow. If the ancestor lives in one area, the descendants both live in that same area. But you can't have a widespread ancestor convey its widespread range to both daughter species. But you can have vicariance, right, where a species divides up into, it's basically splits its range in half. The third method is really a likelihood version of DIVA. It's with some additions. This is probably the most popular method right now. Lagrange is likelihood analysis of geographic range evolution. It has a lot of the same processes. I'll take you through it. This was a paper by Rhee and Smith, this has been cited many, many times. And here, they used a classic example of Hawaiian psychotria, which is a shrub. A subgroup of the shrub lives on Hawaii. And, you can see how this method works. You say, for example, the tips live in Kauai, which is K, or the Big Island Hawaii, or Maui, or Oahu. And you apply your method and retrieve some history that you infer of what happens. And we'll take you through which events they allow in their method. They allow these 3 kinds of events. DIVA just does this sort of sympatric speciation of a ancestor with a narrow range and a widespread vicariance thing. They don't allow this sort of subset speciation where one species buds off within the range of the ancestor. And Lagrange sort of inside of it has basically a big transition matrix. If your possible ancestor ranges are A, B, C, or any combination of those areas, your descendants basically have, you can look at all the possible combinations of descendants. Any cell that's blank in this matrix has a probability of 0, so those events are not allowed. But the sympatry events are allowed, some vicariance events, some subset speciation events. And Lagrange makes the assumption that any allowed event has an equal probability. So you can see on this bottom row, if the ancestor is living in all areas, there are 12 possible ways to split up that range or have a subset speciation event of that range. And each of those is given a conditional probability of 1/12. And this is the model. This is a fixed model that you can't really change. It's just the assumption. Anyone who uses Lagrange is making this assumption. And I think it's often not recognized, really, that that's what's going on. but this is the sort of state-of-the-art model as it exists. It also has these range expansion and range loss events. Like I mentioned, range expansion, range loss. And the nice thing about Lagrange, and one big advantage of it over DIVA, was that Lagrange does take time explicitly into account. The parsimony methods don't really take into account the fact that there's a greater probability of change with a longer branch on a tree. So they do this by, just like any phylogenetic method, you have a transition matrix that describes a Markov process, and you have an increased probability of change as your branch gets longer in time. So the longer amount of time you have, the greater the probability of having a transition is. And so these dispersal and extinction events are parameterized. And in Lagrange, they're left as free parameters. And you input your data, and you run a maximum likelihood search, and you get the optimum values of, or the values of D and E that confer the maximum likelihood on your data. And those are your best guesses at what the dispersal rate is and the extinction rate is. But the cladogenesis model that I mentioned, the vicariance and subset speciation events, that's a fixed thing, and it's not subject to inference. And I mentioned these. These are, and if we look a little more detail at what sorts of events are allowed, the Lagrange allows the events that are in gray here and disallows events that are in white. And interestingly, it makes some different decisions than the DIVA method. Lagrange does have vicariance, for example, but it assumes that one daughter species always lives in an area of one. And it doesn't allow like splitting a daughter species into having one daughter live in two areas and another daughter live in two areas. And that's just probably for efficiency of programming the reason that was made. But that's a much different assumption than the one in DIVA method. And I think it's also not widely recognized. So similarly with subset speciation, the argument was that new species tend to live in small regions, and so we should insist that one new species always lives in a small region. And that's the assumption that's made there. Let's see, and here's that model again. And if we compare Lagrange and DIVA, we can see that somewhat different decisions were made about what processes are allowed. I mentioned that classical vicariance is not allowed, but you still have this sort of vicariance where you split off one area. All right, so we talked about that. A fourth method is this new paper that I was on called Bay Area. It's a Bayesian method. It's basically designed to solve the problem of what do you have when you have lots and lots of areas. Exponentiating a transition matrix gets slow if the matrix gets large. So if you have lots of areas, you have lots and lots of possible combinations of areas. So, and the number goes up at 2 to the number of areas. So if you have, I think if you have 9 areas, you have 512 possible geographic ranges. And you have to calculate the probability of going from every one of those 512 ancestors to every one of those 512 descendants. And that gets to be a large matrix that gets to be slow to do the calculations for. And it gets worse if you have 10 areas, right? Then you're 10,024 by 10,000, or 1,024 by 1,024. And you're pretty soon your programs just crash and you'll never get a result. So this program was designed to solve that problem using a Bayesian sampling technique, and it's really a different talk to talk about it. So it's super cool. It runs hundreds of areas really fast. But one simplifying assumption we made was that there was no cladogenesis process. Cladogenesis meaning speciation. Basically, we assumed that the speciation point on a tree is the same as any other point on the tree, and that nothing happens to the geographic range when you have that. So basically, we assume that this is always what's going on. Or if the ancestor is ABC, both descendant species are ABC. So that's a different assumption, right, than these other programs are making. So a lot of times when you're picking a program to do analysis with, you have a trade-off. You get some advantages for some new technique, but a lot of times you lose some of the sophistication that was in a previous technique. And nobody really has a good sense of what effect this is having on your analysis, or which ones are using the best models. So if we compare all these side by side, the Bay Area thing I just mentioned, these are the only events that are allowed. It's speciation. DIVA allows these events. DEC, dispersal, extinction, cladogenesis, this is the Lagrange model. It allows these events. And you're kind of just stuck picking one or running them all. And typically, often people run all the different methods, and then they get different results for each one, and they sort of shrug and send it off to the journal, right? Like different models, different results. Great. If they all agree, then that's good, right? but a lot of times they don't agree, especially deep down in a tree, which is often mostly what you're interested in. A lot of times you can just look at a tree and say, oh, this whole clade lives in the Big Island of Hawaii. The ancestor lives in the Big Island. But you didn't really need a program to tell you that, right? You're trying to do inference about things you didn't know already. So this is kind of where we're at. Just to give you a state-of-the-art analysis, this is another paper I co-authored, which was the historical biogeography of assassin spiders. Assassin spiders have big, long jaw thingies that they use to eat other spiders without getting too close to them. You know, they have these sort of snap jaws that come around and nab things. And we ran Lagrange and Diva. We sort of did a Bayesian version of those. And the one thing that you can see is that all your tips, you know, the tips and typically the nodes just below the tips, typically they live in one region. And actually all these species just live in one region. But the ancestor you infer lives in basically all the areas or almost all of the areas on either of these methods. So you have this issue of widespread ancestors. Now in this case, we actually did molecular dating, combining that with fossils, putting fossils directly in the tree. So it's sort of a joint and total evidence analysis of molecular data, fossil data, and having the dated fossils directly in the tree. And we inferred ages that are super old, that are sufficiently old. This is kind of a rare case. They're old enough to actually have occurred when the continental plates were breaking up, when Gondwanaland was breaking up. So we thought that was pretty cool because it's actually somewhat rare to do a molecular analysis and get dates that actually are old enough to break up. So we thought there was a reasonable argument for vicariance there. But one thing that bugged us is these methods tend to return the vicariance result, you know, no matter what, right? Wouldn't matter what the dating was or what the sort of ancestral geography was. You would get vicariance almost anything you did. And so it's great if your bias agrees with what you think is going to happen, right? If your method is biased there, that's, you know, you'll get the right answer. But if you want to subject that to test, this isn't ideal. But it was a good learning experience to do this, since we were able to combine the things that hadn't quite been put together before. So these were the assumptions that I reviewed in the methods that exist right now. You know, Lagrange at least doesn't allow true vicariance. You're always assuming you sort of know the current ranges. You don't actually always, but that's a different issue than what we're dealing with now. You're assuming species always start inside their ancestral ranges. And this model that we're using is fixed and unchangeable. And no matter sort of what system you use, you're kind of stuck with that cladogenesis model. So you know, we'd like to know what the right model is. You know, which of these kinds of processes is most important? The one I'm going to focus on here is founder event speciation, which I mentioned, you know, sort of been discriminated against, at least by vicariance biogeographers. It would be nice to put this into the models and see if it does something useful for us, and if it, you know, if it's a better model or not. I'll talk about what founder event speciation is. If you've read sort of the old literature or the neo-Darwinian synthesis literature, Ernst Mayr and these folks from the 50s and 60s, there was a lot of focus on founder event speciation as a population genetics process. So basically the idea is you have a large population. Large populations have a lot of genetic variation, but they tend to, you know, have a lot of basically inertia against change is basically the idea. Whereas a small population, gene frequencies can change quickly. And so the idea was perhaps that speciation mostly happened when a small population dispersed to a new area and got isolated. And when it was a small population exposed to new conditions, maybe morphological change can happen quickly, speciation can happen quickly, and maybe this is a major sort of paradigm for evolutionary processes. This was Ernst Mayr's idea. It got turned into punctuated equilibrium by Stephen Jay Gould and Niles Eldridge and applied to the fossil record, and then consistently misinterpreted ever since then. But anyway, so it's been an important process in population genetics. And amongst certain sorts of biogeographers, it's been an important process. If you talk to people who work on Pacific Islands, they think founder event speciation is basically the most important thing. They think rare colonization events, islands that are basically volcanoes that came out of the ocean, everything that got there had to get there by long-distance dispersal. And a place like Hawaii is very, very isolated. It makes a lot of sense that you would have rare dispersal events, things colonize. They're pretty instantly isolated from whatever the ancestral population was. And whether or not you want to call that speciation right away, it's genetic isolation. And so as a biogeographic process, some biogeographers have always said founder event speciation is important. But it's odd that both certain people, certain vicariance biogeographers have been against it for the reasons we discussed. And whether or not people were sort of against this philosophically, the computer methods that we have don't have it in there. And so people were making this assumption by default, whether or not, if they thought about it, they would agree with it. It's been the only thing they've had. So I mentioned these quotes. Founder dispersal is controversial and may not exist. I'd like to test this, right? I don't just want to make an assumption at the beginning and run your method. You want to be able to test this idea. So all we have to do is, here's the sort of cladogenesis events allowed by Lagrange. If we think outside the box here, we can just add a new class of events. And these are founder events. All you have to suggest is that you have an ancestor, and at speciation, one of the lineages jumps to a new area outside of the range of the ancestor. And there's variations on this depending on how widespread the ancestor was. But all you're suggesting is essentially that one of the lineages jumped to a new area at speciation. And if you take the Lagrange sort of transition probability matrix for speciation events, and you add these jump dispersal events, you can see where they pop up. In some cases, you're adding a fair number of possible events. In some cases, you're not. In some cases, you're not adding any. But it's once you've programmed a Lagrange-style analysis, adding a jump dispersal thing is not very hard. And in the model that I've developed, I make J into a free parameter. So basically, the relative probability of these events is controlled by the parameter J. And if J equals 0, you're back to the Lagrange DEC, the DEC. I call it the DEC model, dispersal extinction cladogenesis. So you have nested models. You have a model that has J equals 0, and you have another model where J is a free parameter. And then we can compare these. So I've implemented this in a couple of R packages. If you sort of think about reconstructing a biogeographic history, I have one package that does the matrix exponentiation, which calculates probabilities of changes along branches, anagenetic events along branches. There's a Clado RCPP, uses some C++ code to rapidly calculate all the different combinations of possible events that can happen at splitting events on a tree. And I stuck this all together in this package, BioGeoBEARS, which I don't think is famous yet, but I have like 8 people on a listserv that are into it, so that's cool, right? This package is the one that you would just go and install if you're interested in this sort of thing from the CRAN archive. You would just go there and do install.packages, and it'll install the other ones. So there's a lot of features. I copied a lot of the features of Lagrange and then added some new features that I always wanted in a program. So you can do things like have distance dependence for dispersal. You can do connectivity between areas and disallow dispersal between areas that aren't connected, and a bunch of other things. And you get an estimate of your parameters, and you can infer ancestral ranges. And there's a couple different ways to do ancestral range estimates that I put in. And here's the URL. I'll put it at the end, too, for people who want to play with it. And this is way too much to absorb, but basically there's a lot of parameters that you can have be free or be fixed. If you implement just a typical Lagrange analysis, you have 2 free parameters, and the rest of these are fixed to these values, and this is what reproduces a Lagrange, a DEC analysis. If you want to do a DEC plus J, right, you want to add jump dispersal, all you're doing is you're changing one parameter from being fixed to 0 to being free. Right? But there are many other models that are accessible from this framework, but testing them all is something that's basically for future work. So if we go back to this Hawaiian psychotria example, and we run it, I did some sort of validation by comparing my BioGeoBEARS implementation to the various implementations of Lagrange in C++ and Python. And there's a lot of numbers, but basically you just look at, is the dispersal rate the same? Is the extinction rate the same? Is the log likelihood the same after maximum likelihood search? And you can see they're basically identical there. And I ran it on a bunch of other data sets, and they basically, I got it working. I get the same numbers. That's great. The one case I didn't get the same numbers was actually a time stratified analysis where you have different dispersal rates at different points in time, which is quite complicated to sort of get right. So in that case, I agree with one of the implementations of Lagrange, and the other ones have minor differences. And so I mentioned we have, if we want to use this to compare models, so I have a Lagrange model with 2 free parameters, and I have BioGeoBEARS version with 3 free parameters, adding this J parameter. As J gets bigger, we have more and more weight, or more and more probability for these founder event speciation events. So when I compared these 2 models, just on the default data set that's used kind of as the test data set for Lagrange, I got a big jump in the log likelihoods that you get. So, and you can calculate use the likelihood ratio test because these are nested models. You can calculate the null, or basically you can test the null hypothesis. The 2 models give you the same, confer the same likelihood on the data. And that's rejected with a p-value of 9.4 times 10 to the negative 8th. You can do AIC and calculate model weights. And the model weight for this particular data set in favor of the 3-parameter model is 99.999 something percent. So that's it's pretty significant for a small data set with 1 extra free parameter, right? So I mentioned these. And the other interesting thing that happens is that the parameter inferences you make are much different. Under the Lagrange DEC model, you get significant probabilities for these dispersal and extinction events. But with a DEC plus J model, these go to 0, basically. And you get a significant weight on these founder events. So ironically, the DEC, dispersal, extinction, cladogenesis, you don't really need the dispersal and extinction things once you've added this founder event speciation. So like I said, this is fairly impressive, but this is just one data set. It'd be nice to look at some more. So I took some other standard data sets that have been used with Lagrange. And you can compare between DEC and DEC-J. You can compare the D values, and often the D values go down to 0 under the DEC-J model. Similarly with E, they often go to 0 or get much reduced. Usually we have significant weight on J on the founder event probabilities. Log likelihoods typically improve either a whole lot or just a fair bit. You can calculate model weights. And in favor of the DEC plus J model, the model weights are anywhere from 30 to hundreds of thousands of times, basically hundreds of thousands of times more probable for the model. And you can do the relative probabilities. And I did a Bayesian implementation of this. Basically use the same likelihood model, but you put priors on the parameters. And you can calculate Bayes factors, and you can calculate log marginal likelihoods and do a Bayes factor. And the Bayes factor I got for the sort of initial data set was 3,500. And if you look at the literature on Bayes factors, they say anything, this is basically a ratio, an odds ratio. Anything above 100 to 1 is considered decisive support for a model, and I got 3,500 to 1. And you can do parameter inference, and in a Bayesian analysis, you get an actual distribution of your parameters instead of just a point estimate. And you can see that J is well outside of 0, for example, when you do the DEC plus J model. So around here is where you should start to get worried, actually, because this looks pretty cool. But what if there's just some weird feature of this model? These are sometimes small data sets. We've added a new process. It's not really tested. Maybe there's some sort of either bug or some sort of inference problem that's leading this more parameter-rich model to work better, and it's not really telling you something about biology. That would be bad. So, and you don't really want to come out and yell a lot about a new model unless you've done a little bit of testing at least. So I ran some simulations under the 2 models and then did inference under the 2 models to see how these things work. So if you take the DEC model and you you run inference under the estimates you would get under DEC, under Lagrange, and then do inference with DEC plus J, which is the more parameter-rich model, you do get, more parameter-rich model will always do a little bit better in log likelihood. But the log likelihood advantage, which is what I've plotted here, is typically just a couple of units, if that. So you add another parameter. The DEC model is nested inside the DEC plus J model. And you get a little bit of improvement in log likelihood, but it's not very significant. Typically, you need at least 2 log likelihood units to get a statistically significant improvement in a supermodel. Whereas if I simulate under the DEC plus J model and then do inference under DEC and DEC plus J, then the log likelihood advantage of dec plus j is huge, right? It's often 10 or more units. And remember, in the empirical case, we had something like 14 log likelihood units. So you can calculate false positive rates and false negative rates for your estimates like this. And basically, when the true model is dec and you run inference under dec plus j, you only falsely conclude that dec plus j is better about 0.05 fraction of the time. And that's exactly what you want. If your p-value is 0.05, you want your false positive rate to be 0.05. So I've basically gotten that. This means that when the true model is DEC, I do not falsely pick this other model, right? I mostly pick the DEC model, which is good. And you get the same result in reverse. When the true model is dec plus j, you very rarely infer that the dec model is the right one. False negative rate is similar. So this is pretty great. This is what I said. You know, this is what got me excited, because this indicates that there are data sets that will favor DEC. It's just that the data sets that we have typically favor dec plus j. They favor the new model. So that is cool. Let me see where we're at here. All right, so we got to move. I should explain what I think is going on. Basically, if you are assuming the Lagrange of the DIVA model, we'll talk about the DIVA model a little bit more in a bit. Basically, you always have to have a range expansion event before you have a vicariance or other event. To get things spread around, you have to sort of postulate some widespread ancestor that then splits up. And this implies that because you have these Markov models explaining this process, this implies waiting times. Every time you're waiting and nothing is changing, basically. Whereas under the founder event model, D and E, those parameters for change along branches, those are 0. So the probability of things not changing is 1. So, and all you have to postulate is basically one founder event right at the speciation thing in this particular case to explain distributions. So if your species tend to live in single areas, and if their configuration is right, according to the progression of islands in Hawaii or something, then you'll tend to get strong support for founder events. So, so I think that's what's going on. And it's useful to ask, does this model choice thing is sort of scientifically interesting by itself, but does it have implications for other stuff? For example, for inferring ancestral ranges, yeah, there are big implications. If you do inference under, this is just that default data set again, you do inference under DEC, and you tend to get some widespread ancestors. This is suggesting that the most probable answer here is that the ancestor of this clade lived on all 4 islands. Whereas under dec plus j, typically you get ancestors just living in one area. And you can sort of see, you just infer a history where you had a couple of jump events to produce the distributions. And if you talk to island biogeographers, or even if you read the original Lagrange paper on this data set, they suggest that this is basically what happened. And it was just the sort of way they built the model, sort of deriving it from DIVA, was essentially they were trying to take time into account and to do this Markov modeling thing. But I think because the whole history of the field has been sort of vicariance based, they didn't think about putting founder event speciation in. And so once you put it in, you do get sort of a dramatically different result in this case. And you can see your sort of confidence in, this is the relative probability of all the different options for ancestral ranges, where white means something living in all regions, and then these different pie slices are different possible geographic ranges. You can see you get some nodes under the dec model that have a lot of uncertainty, whereas your certainty improves a lot for many of the nodes under the dec plus j model. So if all you're interested in is inferring ancestral ranges, you still got to think about model choice, because it matters a lot for what your inferences are going to be and for how confident you're going to be in your inferences. Also, it matters for accuracy. With those simulations, I could check the accuracy, and basically, if the true model is dec plus j and the inference is dec, your accuracy is only 57%, whereas if your true model is dec plus j and your inference is dec plus j, your accuracy is 87%. So if you have the wrong model, you can do much more poorly in terms of your accuracy of inference. We should probably skip this. Basically, your model weights also support this story. And I ran this on a bunch of data sets. Like, we want to know, is this just a couple of data sets, or is this a general pattern? So I picked something like a dozen different Hawaiian and other island clades and ran the model, the weight ratios in the models. And in almost every case, you get dramatic improvement for the DEC plus J model. Mostly it depends on the size of the data set. The bigger the data set is, the more support you get for DEC plus J. There's a couple exceptions, which I don't have time to talk about, but they're basically weird and don't matter. And once you get used to this, the story gets sort of boring. Here's Hawaiian leafhoppers. You infer some nodes as widespread under Lagrange, and under DEC plus J, you get log likelihood improvement of 110 or something, and you infer the ancestor is living. Basically, the blue here is Kauai, which is the oldest Hawaiian island, and that's what you get as the ancestor. And similar story with ancestor probabilities. This is Galapagos lava lizards. And here, they're interesting because you do get improvement in log likelihood, but here, the J parameter went all the way up to 3, which was the maximum value for J. And when J equals 3, that means founder event speciation is the only process being used. And all the other processes have weight 0. So this is a case where the only speciational process going on in this clade was founder event speciation, which is something I don't think you could have inferred through any other method that currently exists. All right, so parameter inference also changes, but we got to move on here. I mentioned how that worked. So there's a couple implications here. One is people should free their minds when they're doing historical biogeography. We're no longer stuck with having one model and just running with it, or running different models and getting different results and just kind of leaving it there. We have a lot of models that we should be exploring and testing. And my inspiration here is just DNA evolution models. The original DNA evolution model was Jukes-Kanner. It was a very simple model that was equal rates of change between all the bases. But it evolved into this GTR model. I don't know if I have a slide, but basically you can make each of these parameters a different parameter. You can have a 5-parameter model or put in additional parameters. And throughout the, from the 80s to the 90s, all these new models were proposed, and eventually you ended up with kind of a super model which nested all these other ones. And I've tried to sort of just jump right to the end of that process and make every sort of process a free parameter. And then it's an empirical question whether or not the data will support having multiple free parameters or not. And that's something that is basically a product for more research. Statistician George Box had this quote, "All models are wrong, but some models are useful." And my main point is we'll never really know the usefulness unless we actually do model comparison. You'll never know if your model is good or bad unless you're comparing it to other models. Let's see. So yeah, here's my beer challenge. For a while, I was getting just everything was supporting the DEC plus J model. And so I wanted there to be some empirical data set that supported the old one. And so I challenged people to buy them a beer if they found one. I actually did find a data set, which was Tagetes butterflies, that supports the DEC model. So I bought myself a beer. And I think I have a slide for that one later. But basically, if you have a data set where many of the species at the tips of the phylogeny have widespread ranges, that's the sort of situation that will support the older DEC model. But if everything lives in one area, it's much more likely to support DEC plus J. And there's a lot more applications here. I'll just breeze you through this. We can do likelihood versions of those other models I mentioned, the DIVA model, the Bay Area model. Now these are all just different parameter values in the BioGeoBEARS supermodel. And we can look at these different models. There's the Bay Area model. Here's how you would change the parameters, but you don't really need to know this unless you email me. I'll send you the slides if you want to do it. This is to produce a DIVA model. You just change some parameter values. To produce a Bay Area model, you change some other parameter values. And then you can calculate this. You could even do a standard discrete character model if you wanted, although I haven't played with it very much yet. The nice thing is you can get likelihoods under all these models, and then you can do model weights and compare. So this is comparing the 3 different models without founder event speciation. So there's 3 colors and 3 models, and the best model for each of these data sets, which is things like Hawaiian Drosophila, Hawaiian silver swords, Hawaiian leafhoppers, all these different Hawaiian groups. The best model, according to model weights, is listed at the top here. And so you get a lot of support for things like DIVA and stuff. When you run 6 models, you do the plus J version of each of these models. This is for non-island groups. When we do plus J versions of these models, which have the same colors but lighter, in the island groups we get basically the plus J models consistent for all these different submodels. They all prefer the plus J version of a model. That's the main message here. Often, and usually that's the biggest signal, like distinguishing between DEC plus J and DIVA plus J, that's kind of a minor thing. You sometimes get a little bit of support one way or the other. But the big improvement comes when you add the plus J to these models. With the non-island groups, I wanted to test this on a non-island system because the island system was sort of expected to have founder event speciation. If you throw in these other guys, non-continental groups, this is where you get the one butterfly group here that supports the DEC model, which is dark blue. So this was when I bought myself a beer. There's another case here that supports another model, but this was like a 5-species tree, which isn't going to have much data to infer anything. And if you look at that butterfly group, right, these are the distributions. These boxes represent how many areas things live in. And there's a lot of species here that are widespread. So if you have a lot of widespread species and sympatry and overlapping ranges, that is the situation where you could use the DEC model and do OK. One other nice feature of having all these inference tools available is we can look at the relative strength of J and non-J, or J in island clades and non-island clades. And basically, island clades, typically the founder event speciation is a stronger process. And depending on how you measure it, it's 2 to 4 times stronger. So for the first time, we could measure the relative weight of these processes in different systems. So that's pretty handy. And you can look at model improvements, and you can see that islands tend to have a bigger model improvement than non-island systems. So there's some conclusions. I think founder event speciation is important almost everywhere. Almost all the data sets falsify the models that are currently in highest use, which I think people should know about, in favor of the plus J models. And with this inference framework, we can finally test different models. Now, there's a lot of implications to this. And I'll try and wrap this up pretty quickly here. Once we're doing model fitting and model testing in biogeography, right, there's a lot of things that you can do that were hard to do or you could sort of only do heuristically before. One is we can actually take another look at the origin of species, like literally the origin of species, right? Either the book or the process, right? Because when you can calculate the weights of these different processes and infer that, you can get a measure of how common are various processes, which is a classic question in evolutionary biology. Like, what are the most important processes producing species in different geographic regions, different periods of time? So we can do that now. Another thing we can look at is trait dispersal correlations. If we think that certain traits that we infer on a tree have an influence on dispersal probability, like flight or flightlessness, we can put that into a model testing framework. So that's pretty cool. One thing that I think has not been realized that I think is pretty important, and I'd love to talk to people about it if they have thoughts about it, is oftentimes when you want to put dates on a phylogenetic tree, you use fossils if you have fossils, but usually you don't have fossils. So if you don't have fossils, your only options are to sort of assume a clock rate that you get from like the Drosophila mutation rate or something, and you apply that to some other insect group and you calculate dates on your tree. That's not satisfactory. The other thing people do is they use biogeographic calibrations. So they say, okay, the big island of Hawaii came up 500,000 years ago, so this node on the tree must be less than 500,000 years old, and they use that to do calibration. And everyone complains about it. Sometimes it's the only thing you can do, but basically people have sort of separated the making that guess from the rest of the inference. But I think we could do it all in sort of a joint framework where we don't assume ahead of time that we know where things were living back in the past. We could just do joint estimation of the dating and the ancestral biogeography, as long as you have some geological history that you can put into it. So I think this should be more fully explored. One big area that needs to be worked on is macroevolutionary models. So I've sort of been assuming that you have a fixed tree where you know all the speciation events. In reality, there's a lot of extinction, and we have a lot of missing speciation events on the tree. And there are some models that are called state speciation extinction models, SSE, that try and take that into account. Really, those should be integrated with the approach I've presented here. If you look at the cladogenesis models in GeoSSE and CLA-SSE, I think basically they're assuming a DIVA model. Basically, there's vicariance, and there's sympatric speciation, and that's it. And as we've shown, that's a huge and strong assumption and probably not the best one. So really, we should integrate these approaches. It involves doing a bunch of ordinary differential equation solving stuff that I'm going to learn here, I think, but I don't know yet. So anyone who's into those, we should chat. And The last thing is species distribution modeling. Let's talk about, we'll skip the fossils here. We can do fossils now, that's good. But if you think about species distribution modeling, if you think about the phylogenetic historical biogeography methods, they take into account phylogenetic information, but they sort of ignore environment. They assume environment's irrelevant. If you look at species distribution modeling methods, these are methods where you take location data and you take environmental layers like temperature and precipitation, and you build a statistical model and you produce a sort of a continuous map of where things live. And this is used, probably used as much as phylogenetics is used. It's like every conservation biologist, land use planner, climate change person looking at species distributions, they're using species distribution modeling. And these take into account environmental information, but usually they're not taking into account phylogeny at all. And this is kind of my main interest here at NIMBioS, is to work on this. So I mentioned standard historical biogeography inference has phylogenies. Distribution modeling, you kind of treat each species as independent, which is a little weird. I think we could build a more complex model. I should probably just move on here and get through this. But basically, we should not, the gray boxes here are things that are fixed, are actual observed data. And really, we're inferring, what the environmental preferences are, as well as inferring a map. And current species distribution modeling, you basically assume species are statistically independent. And if you have a fossil species, you sort of assume that as well. And I think we could do better, right, if we link the historical process of jump dispersal and other events with the evolutionary process of evolution of niche parameters and combine it all into one grand joint inference. That's really what I would like to do. So I think you would get improved inference. I think it would solve some of the problems that species distribution modeling has in terms of overfitting models. You often get a very precise prediction for whatever's living right now, but it's hard to extrapolate it forwards or back in time. So this is kind of the main thing I'd like to work on here, is putting all this together. All you really need is a model for each process, a model that produces the likelihood of the data. And we have models for each of these processes, it's just no one's ever put them all together in one framework. So that's the main thing I'm interested in. I'd love to chat with people who are into that. And I should wrap it up there. People who are interested in looking up more on this or looking up, I've got articles in submission on this process. You can find stuff here, the R package, tutorials, and references to the articles and things. And so I'm happy to chat. My email's on there also somewhere, I hope. You can, there it is, yeah. So so email me or friend me on Facebook or just come chat sometime. I'm just down the hall there. So thank you very much for listening, and I will take questions.